Thank you so much for the presentation here. We got a couple of questions that I think we should start right ahead with them. And the first one is for Professor Zetterberg. What is the role of the amyloid in Alzheimer's disease, and how important is the amyloid in the development of the disease?
Let's see, should I turn on there we have the mic, exactly. This is a very important question. Amyloid it really looks like amyloid is triggering the disease. So it is, but there is a little bit of a debate on in regards to if it's also a disease driver further on. But when new plaques form, it's very clear that surrounding neurons react to the pathology by phosphorylating and secreting tau. So it's an ongoing process. Everything that happens with tau in terms of the biomarkers is amyloid dependent. So most likely, if one wants to go to the core of the disease, it is amyloid that you need to target. One could also think that there might be ways through which you can prevent amyloid from aggregating. But those ways it's a little bit hard to know exactly how to do that, except for changing the stoichiometry of long versus short A beta. So that's why I am so positive to the gamma-secretase modulation approach.
Thank you. Another one for you, Professor. How early can we today predict if somebody is likely to develop Alzheimer's, and how do you expect the future will look like?
Yeah, that's an exciting question too, because now we can now it's data tell us how this could be in a way that I think is reasonable. So we know, for example, a lot about genetic susceptibility of Alzheimer's disease. That has been quite well established now, also in relation to well-diagnosed patients. So there is a possibility now of getting a polygenic risk score for Alzheimer's disease. This is something you are born with, and it will give you your risk from the day you're born until you develop the disease, basically. If one would be very simplistic, it's often enough to look at ApoE epsilon 4, actually. It is a big genetic risk factor for the common form of Alzheimer's disease. So if I think a little bit about myself also with some Alzheimer's in the family, I could in the future then do a genetic test and get my individual risk, and then I could monitor myself with biomarkers in blood when I go to the general practitioner or whatever type of healthcare system I'm in. And then when the biomarkers start to become abnormal, one could potentially initiate a treatment. With Alstatin, we even have the possibility of going earlier. This depends on how safe the drug is. It could be that I know that I have family history of Alzheimer's disease, and then perhaps if the drug is very, very safe and not too expensive, perhaps I could start on the treatment and just make sure that then I could monitor my biomarkers to make sure that the disease doesn't appear in my brain. But this is something that happens in midlife. So this is something which is relevant for the future 45, 50-year-old people. One could also think about it later in life also, but if you want to do primary prevention and try to not get amyloid plaques growing on your brain, this is the age range, 40 to 60, 65, where one should look carefully. If it turns out that the drugs are effective also in when Alzheimer's disease pathology is starting to establish itself in the brain, then it's not that big rush, because then one could look at that at 60, 65, 70. And if it turns out that you can reverse pathology also in symptomatic individuals, perhaps combining it with some type of restoration therapy, then it's also less of a panic. A nice scenario is that established Alzheimer's disease pathology with mild symptoms could be mitigated or dealt with an anti-amyloid treatment, and then you start with something that is that could be a window treatment for 1 to 2 years, and then you would start on something that would prevent the disease from coming back. So the biomarkers work. Okay, the genetic risk can be detected from birth. The biomarkers will react to onset of pathology that most often happens in middle or late midlife. And then the degree of biomarker abnormality will correlate, at least for the phospho-tau markers, a little bit with how advanced and intense the disease process is. And that also works later in the disease process.
Thank you. What role do you think gamma-secretase modulators could play in the treatment of Alzheimer's?
Yeah, I mean, from my point of view, they could play a major role as primary prevention treatments. And if they turn out to be safe and not too expensive, then this could really be something that people with sporadic Alzheimer's disease in the family could, with some luck, this could be a primary prevention treatment for sure. So it could play a major role. And then we also have this more tangible role of evaluating it as a secondary preventive treatment after removal of amyloid pathology with antibodies. So it's quite interesting because then gamma-secretase modulation could have a role during the full continuum of the long Alzheimer process. From before you have symptoms, if you know that you have high risk, and through the early symptomatic and symptomatic stages, and also after removal of amyloid pathology. It might also be that gamma-secretase modulation could make the plaques stop growing, which also could be beneficial. So you have the plaques you have, but they don't continue to spread in the brain. So I think we need some more data on this, but potentially this could be major.
Thanks. Another fun one for you, Professor. What are relevant reductions in amyloid levels when using a gamma-secretase modulator?
It looks from the in vitro studies that you don't have to modulate that much actually. Wow. So it looks like if you just change the relative composition of longer versus shorter forms with 30-40% or so, which is clearly achievable with these compounds, then you really, it's like then you have tipped the balance so that aggregation will not happen. It's pure biochemistry actually, but in humans then it happens in the brain tissue, and we can also do it at the bench, and it really looks like lowering the longer form, increasing the shorter forms. Potentially this could give you more water-soluble heterodimers that are not aggregating further. That could be enough. And this could also be an explanation for why Alzheimer's disease doesn't start early in people, because we are almost managing to keep a beta 42 in solution throughout life, but we fail in the later stage of life, some of us. And modulating gamma-secretase there a little bit to give a different beta milieu, almost a beta composition with a little bit less aggregation prone and a little bit more aggregation not prone peptides or inhibiting peptides that really looks like that could be the magic we are looking for.
Professor Zetterberg, what is your comments to Roche results with their gamma-secretase modulator RG6289?
I haven't looked at the data carefully, but what I've heard, it looks really promising and supportive of the approach.
Thank you. Moving on to the subject Alzecure a little bit more. And this question is for you, Johan. What are the potential advantages and differences of a gamma-secretase modulator versus gamma-secretase inhibitors and versus base inhibitors?
Yes, as I mentioned earlier on, I think it's a very different mechanism of action in that we don't inhibit the enzyme and thereby not inhibiting the cleavage of other enzyme-related substrates such as Notch. So it's certainly a much safer mechanism of action, specifically than targeting the ratio, as we heard Henrik mention earlier on, the ratio between the shorter and the longer forms. That's the major focus of this mechanism of action, and not inhibiting enzyme activity whatsoever. So it's a very different mechanism of action, clearly with a safer, much safer strategy to move on with.
Thank you. And this is one for you, Johan, as well. What are the potential advantages and difference of a gamma-secretase modulator versus a antibody?
Well, I think that's again relating to you have the differences between the small and the large molecules. With the small molecule compounds such as alstatin, which have the advantage of being small and actually going through the blood-brain barrier much easier, reaching the target. So that's clearly an advantage. Also, you can take it as a pill, as I mentioned, as an oral treatment, which certainly helps a lot with the patients not needing to get to hospital. And it's also much more cost-effective to produce. So certainly, I think given that so many million people are currently living with Alzheimer's disease, there's a huge population that needs to be treated. So a cost-effective treatment, I think, is going to be crucial to reach a larger population. Obviously, there will always be a niche for antibodies. That's clear. But I think what we'll see more and more is actually combination therapies. We've seen here now maybe together with gamma-secretase modulators and an anti-amyloid antibody as a maintenance therapies is maybe something we'll see as well. But I think, again, as Henrik mentioned, that gamma-secretase modulators could also be used at a clearly earlier stage, thereby being able to use as kind of across the disease continuum. I think is really interesting concept actually with the gamma-secretase modulators.
Thank you. Professor Zetterberg, what is your view on combination therapies, for example, GSMs and AB antibodies? In what ways could they be combined?
I think there's a huge potential for this, because now we don't really know how to deal with the anti-amyloid antibodies. First of all, they're not approved yet in Europe. That's a problem. But in the US, where I also work, of course, patients are getting these treatments now. But no one knows really when to stop. So should one stop when a patient has gone amyloid negative according to PET scans, or should one then reduce the dose and continue to infuse these antibodies but at the lower dose? Or could one, because one problem with amyloid diseases is that misfolded proteins have a tendency of coming back. That pathology has a tendency of starting again. So one needs to study this scientifically, of course, but removing amyloid with an anti-amyloid antibody, and in the later phase of this treatment, for example, during the last 12 to 18 months, perhaps starting up on a gamma-secretase modulator and then stopping the antibody and maintaining the patient on an oral anti-amyloid aggregation compound in the form of a GSM. To me, it feels very. I really like that thought, and it makes very much sense to me. So we need to do those types of studies, I think.
Thank you.
So, potential, I think.
Thank you. Johan, what patient groups could potentially benefit from a gamma-secretase modulator treatment?
Now, I think, as I highlighted, I think the two populations that we would single out as kind of the first way in would be as a maintenance or combination therapy in patients that are being treated with anti-amyloid antibodies, either as a maintenance therapy, so after they've finished their and reached amyloid negativity, so to prevent the buildup of new amyloid, I should say, as a combination therapy together with anti-amyloid antibodies to prevent both the kind of formation of amyloid and at the same time clearing existing amyloid pathology to maximize the effect of the combination approach. Both of them are clearly ways forward based on what's currently out on the market or on the way to the market. As a long-term possibility, clearly prevention, as we heard from Henrik, I think is a really exciting possibility. Based on its mechanism of action, this could clearly be a very interesting preventive therapy for the disease. Because again, if you go in as early as you do with the preventive therapy, you don't need to lower A beta that much because you do it under such a long time that you would expect that a low dose of a gamma-secretase modulator would over time have a very beneficial effect and actually could prevent the disease from developing. And that would also, I mean, a low dose of a gamma-secretase modulator would also then have a much even better safety profile because it's so low doses. So I think it's also a very interesting concept. But again, we would start with maintenance and combination therapy.
Thank you. When would it be optimal to start Alzstatin treatment if biomarkers and infrastructure is in place?
I think that's for Henrik.
Yeah, I think that biomarkers will be in place in most parts of the world within the next few years, actually, through the blood tests. And so I think actually the biomarker side will almost, that will not be a problem, basically. That will be, so now it's about moving these trials forward. That's what I would say.
Thank you. Here's a question for you, Martin. What is Alzecure's business development strategy with regarding to licensing? Sorry.
Our strategy is to out-license 1 to 2 projects to finance our others. And our main strategy is to out-license our pain projects. And as you saw earlier in this seminar, we have just had positive phase 2A data on TRP1 antagonists. So that would be very suitable for out-licensing. Then we also have a Trk-PAM, another pain project, which is also in a good position. So to out-license primarily the pain projects to finance our Alzheimer's projects, that is our main strategy. Then, of course, we are in dialogue with various companies also on our Alzheimer's programs. So depending on the potential partner and depending on the setup, we are also open to partner our out-license on those. But the thing there is also, we have talked much about the gamma-secretase modulator program, Alzstatin. To drive a project like that isn't very costful. What we are preparing for is a SAD/MAD, and here we also expect to have proof of mechanism with showing how we can lower amyloid load, the production of A-beta 40/42. And to be able to do that and then really to generate that data would be a great value driver, which we are very keen to be able to do ourselves. And we already have done several SAD/MADs in the company, so we know we are very capable of doing that. So to go back to the question, our focus is to out-license our pain projects to drive our Alzheimer's projects.
Thank you. Another question for you, Martin. What will validate Alzecure's gamma-secretase modulator and creates reasons to believe?
I think that it's the phase 1 where we have shown how we can lower the production of A-beta 40/42 and also increase the production of the potential beneficial forms, A-beta 30 and 37 and 38. And then I think we have in a good way validated, but of course we then later on need to take it into the patients, but also as Henrik has talked about before, I mean, the patients who can be relevant to treat later on are not the patients who have cognitive deficits, etc. It's based on the production of A-beta 42 and then also the risk factors. So that's why it's very interesting to define who's the patient and how do we look at this. So that's a very interesting question. But here we also think that the FDA and EMA will be very open to look at the biomarkers, and hopefully based on this we can use Alzstatin just as a statin, as Henrik talked about before. So interesting future.
Thank you. We have 2 questions left here regarding new restore, and the first one is for you, Johan. What is the support of the role of TrkB, BDNF in the cognition?
Well, there's plenty of support actually supporting the role of BDNF in cognition. So BDNF plays a really important role in synaptic plasticity in the hippocampus, which is the really important relay station in the brain, handling and putting together a lot of sensory information that the brain receives. So it really is in the key place of where cognitive processing is taking place. It's also known that BDNF, for example, in the Val66 met polymorphism, so individuals which have about a 30% decrease in BDNF release, there you see that these individuals, when they do develop Alzheimer's disease, you have a much more rapid cognitive decline. So clearly indicating that this system is crucial for normal cognitive function, but also plays a really important role when it comes to sustaining cognitive function during pathological load. So it's clearly there's very much publications in around the role of BDNF in cognitive processing in synaptic plasticity as well.
Thank you. Moving on to the last question here, Martin, it's for you. How does Alzecure's Trk-PAM differ from ESI's Trk-PAM?
So ASI, as you say, have a so-called Trk-PAM, and what that means is that they are increasing the signaling of NGF, and NGF is key to brain health. We also have this with NeuroRestore, but what we also have is we are increasing the signaling of BDNF, working on the Trk-B receptor, and BDNF is key for the cognitive functions to improve learning and memory capabilities. And then also what we have seen from preclinical work is that our molecule can also, as it looks, have an effect on as an antidepressant. So there are several benefits and differentiation points.
Thank you so much. This was the last question I had, so now I will leave over the word to you, Martin, for some concluding remarks.